Mitosis is a form of cell division that results in two identical daughter cells with the same number and kinds of chromosomes as the parent cell. The stages of mitosis are listed in order are as follows: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. We will first look at interphase, or the growth phase. Interphase is not a part of mitosis, but includes the G1, S, and G2 phases of the cell cycle. During each of the G phases, more commonly referred to as the growth or gap phases, the cell is constantly increasing in size and synthesizing proteins. During the S phase, the chromatin, or uncondensed DNA in the nucleus, which is contained within the membrane of the nuclear envelope, is duplicated through the use of multiple proteins. Near the end of interphase in animal cells, two centrosomes form by duplicating a single centrosome. Each of these centrosomes are made up of a pair of centrioles, and their purpose is to organize the microtubules in the mitotic spindle. Also part of the cell during interphase is the nucleolus, which is a small section of the nucleus. Prophase is the first stage of mitosis. During prophase, the chromatin in the nucleus condenses into units called chromosomes, which are each made of two identical sister chromatids conjoined at the center, forming a centromere. In this step, during prophase, also in the nucleus, the nucleolus disappears. In the cytoplasm, the centrosomes begin to form a structure called the mitotic spindle, which is made of microtubules connecting between the centrosomes in a locking hand-like shape. Also around the centrosomes, an array of microtubules forms. This is called an aster. In between prophase and the next phase, metaphase, there is prometaphase. In this step, the nuclear envelope, which previously held all the chromosomes in one area, fragments and disintegrates. The microtubules of the mitotic spindle also start to extend as the centrosomes move to other sides of the cell. They move to the opposite side. The chromosomes also condense further, each having a kinetochore on each sister chromatid for the mitotic spindle to attach to. Some of the microtubules attach to the kinetochores on the sides of the chromosomes and move them around, turning into kinetochore microtubules. Others, named non-kinetochore microtubules, connect to each other from both ends of the cell. Next is metaphase, which is the longest stage of mitosis. In metaphase, the centrosomes are located at opposite sides of the cell, each having a kinetochore microtubule attached to a kinetochore on each sister chromatid. In this phase, the chromosomes are lined up in the center of the cell along an imaginary plane called the metaphase plane. The mitotic spindle is also reaching across the length of the cell in this phase. Just after the longest phase, metaphase, we have the shortest phase of mitosis, which is anaphase. In this phase, the two sister chromatids are separated from each other by the proteins making up the centromere being cleaved, and each is now called a chromosome. The kinetochore microtubules assist in this process by shortening, but remaining attached to the chromosome. Also during this phase, the cell elongates in the direction perpendicular to that of the metaphase plate. Each side of the cell is left with an equal number of chromosomes after anaphase. After anaphase, the cell quickly moves into telophase. During telophase, two different daughter nuclei form around the chromosomes at the ends of the cell. Both of these nuclei have identical sets of DNA contained within them.
Isn't it harder? Is it harder when you Why does he have this ring?
I have a reputation
Following telophase, the cell undergoes cytokinesis. During cytokinesis, the cell splits by dividing the cytoplasm in two. This is done along the cleavage furrow in animals, which is located along the metaphase plate and is used to pinch the cell into two. In plants, this is done using a structure called a cell plate, which is used to form a new cell wall. Done.